Hey guys, today we're going to be talking about meiosis, which is another cell division process that sets the stage for sexual reproduction. Now we've already talked about mitosis, which is in charge of making somatic cells for healing, growth, and development in asexual reproduction, but today we're talking about a completely different process called meiosis. So the first thing I want to do today is I want to define what is reproduction. The definition of reproduction is anything that results in the creation of a new organism. And we've already talked about one type of reproduction, which is asexual reproduction. And that's where we make a new organism from only one parent. And because we're only getting genetic information from one parent, those offspring are identical to the parent and all the other offspring. And this means a few things. We can look at some pros and cons that come along with that. Some benefits of asexual reproduction is that it is very fast because it's a simple process, because we're only going to one organism for that genetic information. Because the organism doesn't have to look for a mate, that saves time and energy from courtship behaviors and things like that. And because this process is fast and we don't need a mate and it's relatively simple, we can produce a large number of offspring. Now, producing a large number of offspring can typically be a good thing because it ensures the survival of the species. Even if a few die off, you still have plenty in the population. Now, large offspring can also be a bad thing because if we overpopulate, what that's going to do is it's going to create an unneeded and unnecessary amount of competition. And the more competition you have in the area, the more the population will decline. Now also another bad thing, and perhaps the worst thing about asexual reproduction, is this no genetic variation. And what that means is if something changes in the environment, maybe there's a new virus introduced or there's a new predator, something like that, then whatever weakness that organism has, all the other organisms of that species have it because they're all getting the genetic information from one parent, so they're all the same. So if that virus or that predator has locked on to that one variation and all of the population have that same variation, then that's going to be bad for them. So you could potentially wipe out your species like that. And also evolution is very slow because there are no variations, and we'll talk more about variations and natural selection later on. Today what we want to talk about is sexual reproduction. And sexual reproduction is just, once again, making a new organism by combining genetic information from two different parent organisms. Now take special note of this definition because this is going to be important later on. A lot of students have a misconception about sexual reproduction. It also says that offspring are genetically different from parents and other offspring.
Sorry for the interruption, but I need to kind of explain how break is going to go. Mr. Charlie delivered your box to you this morning with your snacks. Candy is $1. All the chips are 75 cents. We do not have drinks today. We will have drinks. They're just back ordered right now. So go ahead at the beginning of break, when the bell rings for break, sell your snacks. There will be a teacher out on the hallway with a money box if you need change in your classroom. They're going to be delivering you a Ziploc bag. Write your name on that bag with a Sharpie. Put your snack money in the bag, and the teacher on the hall will pick up all of the money bags from the teachers that are selling snacks before break ends. Thank you.
Okay, and we're back. So one thing about the organisms for sexual reproduction is that the offspring are going to be genetically different from the parents and the other offspring. For instance, you look different than your parents. You might have some similarities, but ultimately you're not an exact clone of them. And same thing for your siblings. This means a few things for sexual reproduction as well. We can always look at the benefits and the downfalls of this. So the benefits of sexual reproduction is that variety. That is so important. It's probably the best feature for sexual reproduction. Because we have variety as a species, that speeds up evolution through natural selection, which we have a whole unit on later on. But some bad things about sexual reproduction is that it costs a lot of energy. Not only do you have to find a mate and fertilize the egg, but you also have this gestation period for some organisms that do internal fertilization, and that's just the pregnancy time. On top of that, most sexually reproducing organisms are more complex, and there's a lot of parental care there. So whereas asexual reproduction, you might have bacteria that don't necessarily take care of their young, sexual reproduction, like humans, kangaroos, things like that, they're going to take care of their young typically until a particular time period or until that organism is mature enough to live on its own. Now once again, if we look back at our definition for sexual reproduction, it says that a new organism is formed by combining genetic info from two parent organisms. It's very important to know that this does not always involve the act of sex. So not all reproduction involves sex, or not all sexual reproduction involves sex, and also not all sex results in reproduction. This is the biggest misconception every year with students in sexual reproduction. The key to sexual reproduction isn't sex, it is fertilization. So you might ask, okay, so what is fertilization in? Fertilization is the uniting of egg and sperm. Egg being the female gamete or the female sex cell, and sperm being the male gamete or the male sex cell. Once these two things come together through the process of fertilization, that creates a zygote, which is the first cell of a new organism. And that has the full number of chromosomes, which we'll talk a little bit more about later. So fertilization can happen many ways, but the main two categories are internal fertilization, where the sperm fertilizes the egg inside the female's body, or external fertilization, where the sperm fertilizes the egg outside the female's body. Obviously, with humans, we use internal fertilization. However, with things like fish and things like that, that's typically external fertilization. So there's an issue here, though. If we take an egg and we take a sperm, and those egg and sperm have 46 chromosomes, we're not going to end with the 46 chromosomes that we need for human somatic cells, right? So this is why meiosis is so important, because it doesn't create these somatic cells. What it does is it creates those gametes. So meiosis is a process of creating gametes from an original somatic cell. So we take a somatic cell, which is our regular body cells, anything but egg and sperm that have 46 chromosomes, and we divide that cell into 4 daughter cells with half the number of chromosomes. What those cells are called are gametes. And gametes are haploid cells, meaning that it has half the number of chromosomes of the original cell. Examples of this are egg, which are the female gametes, and sperm, which are the male gametes. These are all known as sex cells, so the other name for gametes are sex cells. And gametes have to be used for fertilization and sexual reproduction. So you have to have an egg and you have to have a sperm for fertilization. Now what that means is your egg and sperm have to be those haploid cells with half the number of the original chromosomes of the cell. So instead of having 46 chromosomes in a human egg, we only have 23. So that when we combine it with a male gamete or male sperm with also 23 chromosomes, that adds together to give our regular 46 chromosome count for a human somatic cell. So once again, it's all about the number of chromosomes, and that's what makes meiosis so important. Now, sexual reproduction, we talked about the most important thing about it is variation. And that variation happens in 2 different ways. The first form of variation comes through fertilization, which is just combining genetic info from 2 different parents. So obviously you're going to have some genetic variety there. The other form of variation comes from crossing over, which is when we're creating those gametes, all those gametes that are formed are actually going to be genetically different. We'll talk more about that in a moment. So if we look at fertilization, we're taking an egg from a female and a sperm from a male, and those egg and sperm have different information for traits. So like, let's say the male has coded for blonde hair, the female has information for brown hair. When we get to the child, it's going to have information from dad and mom for hair color. So it's going to have a trait, or it's going to have a chromosome, a gene for blonde hair and brown hair. Now it might express blonde hair because let's say that blonde hair is dominant to brown, which it's not, but it's only going to express one trait, but in its cells, it's going to have the blonde hair and brown hair. Same thing for the height. If mom is tall and dad is short, and they both give those genes for that trait, then the child, the baby, is going to have information for tall and short height, but it's only going to express one of those. And we'll talk more of that when we get to our genetics unit. But it's just important to know that the child is going to be different than the mom and the father. So the other form of variation comes with crossing over. And crossing over is the process that occurs in prophase 1 of meiosis, in which homologous pairs exchange genetic information. This results in the creation of 4 genetically different daughter cells. And we're going to look at this process. So to begin meiosis, we just start with a regular somatic cell. And somatic cell just means that we have 2 chromosomes, one from mom and one from dad, for a specific trait, right? So when we're about to divide our cell, just like in mitosis, we go through this process of interphase, where we take those chromosomes and we duplicate the information to make those sister chromatids. Now what's different about meiosis is those chromosomes, they code for the same things. Like this is coding for hair color from dad, this is coding for hair color from mom. And so since they have the same genetic information in the same order, we can line them up right next to each other. And once they're lined up, they can actually swap information. They can exchange some of that genetic information. This is called genetic recombination. And this happens in prophase 1 of meiosis. And this is what makes the genetic variation from meiosis possible. So just like mitosis, we go through prophase 1, and then our chromosomes, they line up in the middle, and we have our centrioles that are going to attach spindle fibers to the centromeres of the chromosomes, and they're going to rip them to other sides of the cell on anaphase 1. Now telophase 1, we're going to reform our nucleus around those chromosomes, and we're going to divide the cell. Now, another difference between mitosis and meiosis is we do PMAT 2 times. So now we're going to start the process all over again. So these are 2 cells from meiosis 1, and now we're starting into meiosis 2. So that's why you see the 2 behind the phase name. So we've started, we have our genetically altered chromosomes here, and now we're about to go and divide them again, because remember, our goal is to make cells with half the number of chromosomes as the original cell. So we go through prophase 1, then those chromosomes line up in the middle, and keep in mind you would have 46 different chromosomes lined up. This is just showing one at a time. So our centrioles are going to attach our spindle fibers to the centromeres again, and they're going to rip them to alternate sides of the cell through anaphase 2. Once we reach the end of anaphase 2 and go into telophase, we're going to start splitting those cells and reforming our nuclear membrane. And by the very end of meiosis 2, you will end with 4 haploid daughter cells, meaning that they had half the number of the original chromosome. Now, if we took all of those cards and we smushed them together, this is essentially what it would look like in a summary. So we had a parent cell with 46 chromosomes, which in this case we only drew 2 chromosomes, and we ended with 4 daughter cells that are haploid cells. So we drew 2 in the beginning, so we only ended with 1 chromosome in each cell. So if we were talking about human cells, if we had 46 in the parent cell, after meiosis we would end with 23 chromosomes. And this once again allows for that fertilization where we have 23 chromosomes with the egg, 23 chromosomes with the sperm, and they combine to give you the 46 in a zygote. So this is the process of meiosis, and we'll go over a graphic organizer here in a little bit to give you a full rundown on one sheet of paper if that helps you out a little bit more. If you have any questions, let me know, and I want you to have a great day.